Thank you, Gabby, for the introductions. Thank you all for coming and joining us today. It's really great to see such a wonderful full room of everybody, and I'm sure you're going to learn a lot about the great science programs we have going on at ALS TDI today. Thank you for all of our staff, marketing group, development group, for organizing the meeting today, tomorrow, yesterday. A lot of work goes into pulling these 3 days off for our entire staff and team, so thank you to all of you. So Fernando gave me the task of kind of giving a high-level overview of some of the things that we're going to touch on today, so I'll do the best I can. If you have any questions, I'm happy to answer them at the end. And I have no idea how to make this go forward, so let's try. There we go. So the typical first slide for those of you in the room who might not know what ALS is, which is probably pretty unlikely. The classical definition, amazingly enough, really hasn't changed that much over the years. It affects both the upper and lower motor neurons. A diagnosis, as most of you know, is a diagnosis of exclusion, which is quite challenging. In good metropolitan areas, it can be a fairly short diagnostic process. Unfortunately, in other places, it can end up being quite long, especially if your primary care physician doesn't happen to see a lot of ALS patients. You'll go through this typical thing of maybe it's a peripheral neuropathy, maybe it'll go away, why don't you come back in 3 months? As far as standard of care, that's improved quite a lot in the decades since I've been at ALS TDI. Median survival is probably not really 3 to 5 years anymore. It's definitely increased out towards the longer edges of that. But with that said, we still don't have that disease-modifying therapy that all of us in this room want. We have 2 FDA-approved drugs, Riliatec approved decades ago. Lots of data to sort of, there's some marginal improvements, but again, not what we want. And then in 2017, MT Pharma got Radicava approved here in North America. They've done a great job launching that and making it more widely available. The insurance companies have finally got behind it. And we're kind of still waiting to see what really the long-term effects of Radicava might be in our community. But again, I think by now, if it was really this massive disease-modifying drug, we all would be much more excited about what we think we're seeing in the clinic. ALS comes in a couple of flavors, and I'm going to be very generic here and not try to be too broad, but we call some forms of ALS genetic, small percentage, 10%, maybe a little bit more, tends to track through the family. You're inheriting a gene from one of your parents. C9orf tends to be the most prevalent one that was identified most recently. SOD is probably the one that most people know about because it was first identified in 1997. One last take-home message I'll say about that, because I think what you're going to hear from our science team during the day today is to understand that knowing the cause or the genetics of a disease is not a linear path towards an effective treatment. If you think about what I just said. SOD1, we've known about it for 30 years. We still to this day don't have an effective treatment for the 2% of people with an SOD1 mutation. So lots of folks think that as soon as you know the genetics of a disease, that's going to translate right into an effective therapy, and that's not always the case. Is there promise on the horizon that we're going to shorten that timeline? Yes. Biogen has a couple of fantastic potential clinical candidates that are in phase 2 that are antisense oligonucleotides that, for instance, target SOD1 or C9orf. There's companies bringing gene therapies forward as that technology platform has matured that are also potentially going to accomplish the same thing. But for sporadic ALS, which is 90% of people that get diagnosed, we don't really understand what the underlying genetic component is, which makes the drug development process a little bit more complicated. But much like a disease like multiple sclerosis, where the genetics is a little unclear, there's 14 FDA-approved drugs for multiple sclerosis, and some of them are fantastic. And I think in ALS, we're getting to the point where we're going to start mimicking some of the great things we've seen in diseases like multiple sclerosis, psoriasis, where there's plenty of effective treatments for everybody. Which leads us to the complicated slide that I'm not going to go through. So ALS is a complex disease, and I hate to be redundant and use that word, but for scientists in the room, any single pathology or molecular mechanism you can think of, which are the fancy words on this slide, you could Google search it or PubMed search it with the word amyotrophic lateral sclerosis, and it's going to come up. There's components of misfolded proteins, there's neuroinflammation, there's neuromuscular junction occupancy issues. You don't have to understand the words, but the good part of the fact that there's lots of different biological things going wrong is there's lots of different opportunities for drug development. And lots of companies right now are tackling various mechanisms that are on this particular slide. So a quick schematic on drug development so that people can kind of at least understand the lingo a little bit. So preclinical drug development, which is somewhere up on the slide right here, drug discovery. The first thing when we think about drug discovery is understanding the disease. It's awful hard to make a drug for a disease if you don't understand anything about it. So as scientists or clinicians, we use tools for the most part. If we can make animal models of the disease or cell-based models of the disease, if we can bring in people with the disease and collect specimens from them, blood samples, biopsies, and cancer biopsies are obviously the most popular. That helps us understand what might be going wrong, all the fancy words on that last slide. That then leads to, okay, if I can fix this part of the disease that's going wrong, maybe I can identify a lead candidate, a drug that might potentially slow the disease down or even cure it, for instance. That's what we think about when we talk about preclinical drug development. It's a long process. You have to study the disease. You have to screen lots and lots of potential drugs. You'll find some lead candidates that look interesting. You'll move them into animal models, do a lot of work there to show that the drug does what you think it's doing, demonstrate that it slows down disease, understand how to dose it most effectively. Complicated process. The next part of drug development, which most people love to ignore, but it's actually really hard and gets incredibly more expensive, is once you have a lead candidate that looks like it works in the lab, you haven't solved everything that the FDA and the patient community wants. Can you actually make enough of the drug? And that sounds like a trivial problem. It's not. IND-enabling safety studies. Just because the drug worked in a mouse doesn't mean it's going to be safe in people. Typically, you have to test the drug in multiple different species, including in some cases monkeys. Incredibly expensive. There's the whole regulatory process of getting the drug approved and demonstrating you have all of the data. So it's a complicated process. Finally, you get into the clinic. The first study is not going to address whether the drug slows the disease down. It's still about safety. You have to understand whether the doses that you want to bring into your target population are actually safe. And then finally, we get to the best part of the drug development process that all of us in this room want, and we all want it to move faster, is phase 2 and phase 3. Proof of concept studies, test multiple doses, hopefully have some good clinical endpoints to measure to show that the drug is actually working. And in ALS, that's not an easy thing, as you'll probably hear in a couple more of my slides, but probably throughout the day. And we need to improve on that process. But ultimately, at the end of the day, if you can get to the point of phase 3, the safety looks good, you've got great clinical endpoints, and you're slowing down the disease, the FDA is really behind us getting things to work on a disease like ALS. But there's a lot of check boxes along the way. So historically, there used to be this big wall in the pharmaceutical industry, and I worked at Herx Marion Cell for a lot of years, Aventis, and this was a problem that is odd, but it was what it was. Years ago, the pharmaceutical industry did not engage the patient community like you see today, and we kind of take it for granted today, but when I was working in those organizations 30 years ago, I'd never remember meeting any person with any disease that the company was working on, and we were working on 40 disease indications. That's a pretty remarkable thing that the infrastructure of the pharmaceutical business didn't talk to the community. And that's a great thing that has evolved in the last 20 years. So how has that impacted the process? So we don't really think of it as a linear schematic like was on the slide a couple of slides ago. It's more of an iterative process, which is now, I've drawn circles for all of the little processes. And even a short time ago, the patient community was only involved in the last steps. We started to really want to collect biospecimens because we could sequence genomes and look for biomarkers in tissues and stuff. And we thought that that was fantastic. But in the last, 5 years or so, there's been even a more dramatic paradigm shift where that second half of the process, the clinical trial part and the discovery part, is becoming so incredibly integrated with tools that we didn't even have a decade ago, that it's really changing the way that scientists and clinicians can start to think about how do you look at a disease from a patient community, translate and bring that alongside with all of the animal models and cell-based models we're using in the lab, and bring all of those tools together in fancy words that we call translational research and precision medicine. And those two things are a little bit intertwined. The translational research piece is how do you bring the patient community into the lab and utilize that information, both clinical, biopsies, genetic data, to build better tools in the lab to find your targets and get through lead discovery quicker. On the back end for precision medicine, how do we target the right drug to the right subgroup of patients that might respond? All of this is what we're going to be talking about today. And TDI had a lot of insight into how important that would be. In 2013 or so, before anybody was talking about precision medicine and ALS, we decided to hire a couple of scientists, Matvey Lukashev out of Biogen, committed to him of setting up a team to do translational research and start to leverage these new technologies. Fernando and our team sat in a room and said, precision medicine, they do it in cancer. I think it's ready for prime time in ALS. Let's do it. And it started off as a small program, and today we have the biggest precision medicine program in the world. We have a fantastic collaborator in Google because the analytics of this much data is really tough. And what I'm hoping you're going to hear today is how we're starting to leverage all of those things to get to effective treatments and a cure for disease like ALS. And this is what a precision medicine program can look like. For every person that enrolls, first of all, the most important part of our program, we share all of the data with the users. If you enroll as a person with ALS, we're going to share all the data live in real time. We're going to stick you for blood samples and things because we want to sequence your genome. We want to track biomarkers in blood over time. We want detailed history, not just when you got your symptoms and the typical things like age and gender. We want to know your lifestyle. Did you play sports? Were you in the military? How much exercising have you done in your life? It's a very complex program. We want you to give us monthly data points around how your voice is doing, and you'll hear some more about that today. Try to wear devices on your ankles and wrists, accelerometers that are hopefully more quantitative than ALSFRS, which is the standard endpoint that we put in trials, and really aggregate all of that data together and bring some of your specimens into the lab so Matvey's team can actually utilize our new genomics tools and platforms to help push leads forward quicker than we ever could before. And you'll hear hopefully lots of stuff about this today and in the poster session at the end of the day, which is going to be fantastic. And on that note, I'm going to thank all of you as people with ALS that have contributed to programs like the Precision Medicine Program and for engaging us at events like today. This is your day. We're here to answer questions for you, so don't be shy.